Hi everyone, it's Dr. LeBlanc with Unit 10A, Mitosis, Meiosis, and Mendel. Our learning goals for this lecture are to understand the difference between haploid and diploid cells, to understand what centromeres and kinetochores are, to understand that chromosomes can have either one or two chromatids, to understand the different stages of the cell cycle, to understand the structure and behavior of chromosomes during the cell cycle, to understand the difference between mitosis and cytokinesis, to understand the behavior of chromosomes during mitosis and during meiosis, to understand genetic recombination and crossing over, and to understand the stages of meiosis in gametogenesis. This is a short video showing mitosis in a plant cell. Here you can see the chromosomes are already condensed, and you can even see two sister chromatids in some of them attached. The centromeres are going to line up on the metaphase plate, and the microtubules will attach to them and pull them to opposite poles of the cell. You're getting ready. Here they go. The two chromatids separate, and each set of chromosomes goes to the opposite poles, and then a cell wall forms in between. You can also notice that the chromosomes are now decondensed back into chromatin. Eukaryotic chromosomes are inherited in sets. Most species are diploid with two sets of homologous chromosomes. Humans have 46, 23%, dogs have 78, or 39%, and fruit flies have 8 total chromosomes, or 4 chromosomes per set. This is a schematic drawing of a single chromosome with two sister chromatids as it exists after the synthesis phase in G2 of the cell cycle. So this is a pair of sister chromatids. Remember, a chromatid is one long strand of DNA from the tip of the P arm to the tip of the Q arm. The DNA at this point where the two chromatids are joined together is called the centromere. The centromere is the DNA that's hidden beneath a kinetochore, which is a set of proteins that attaches to the centromeric DNA. Kinetochores are responsible for attaching to microtubules and pulling the sister chromatid apart, one each to each pole. So the inner plate contacts the centromeric DNA, the outer plate contacts the kinetochore microtubule, and the middle layer contacts the other two. Okay. This is a human karyotype for an XY individual, genotypically male. If we look at one homologous pair of chromosomes up close, we can see that these two chromosomes are a pair of homologs. Okay. If we look at one of these chromosomes up close, we can notice that at this stage of the cell cycle, the chromosome exists as a set of sister chromatids that are very tightly held together. They're not just held together at the centromere as is usually depicted, but in fact they are tightly juxtaposed to each other by a complex set of proteins. Sister chromatids are attached to each other at this phase, and this is one chromosome, one single chromosome with two chromatids. So we're going to look at the behavior of chromosomes during first the mitotic cell cycle. The mitotic cell cycle is comprised of different segments: a gap 1, a gap 2, the synthesis phase, and these are often referred to by single initials, G1, S, G2, and then of course mitosis itself, which can be subdivided into different phases. Cells that are not participating in replication and proliferation are somewhat quiescent. They are said to be in the G0 phase. The term chromosome is not equivalent to chromatid. In G1, after mitosis, a chromosome is a single chromatid. But after the S phase in G2 and early M, a chromosome is a pair of sister chromatids. So at the end of S phase, the cell has twice as many chromatids as chromosomes. For example, a human has 46 distinct chromosomes in G1 and 46 pairs of sister chromatids in the S and G2 phases. If you draw it, you can understand it better. Let's look at a schematic of just those events. Here is a cell that has two pairs of homologous chromosomes. There are 1, 2, 3, 4 chromosomes here, two pairs. This is one pair of homologs. This is the other pair of homologous chromosomes. After S phase, these chromosomes now have two sister chromatids because we've replicated the DNA in that original chromatid. They are held together at the end of G2, the chromosomes have condensed. During mitosis, these sister chromatids are going to separate from each other and produce two new daughter cells with the same genetic composition as the original mother cell. The G1, S, and G2 phases of the cell cycle are often referred to as interphase as opposed to mitosis, and mitosis itself can be broken down into prophase, metaphase, anaphase, and telophase. This is a homologous pair of chromosomes. One presumably came from the mom and the other from dad. The genes are aligned along the chromosome in specific order. Here is depicted 3 genes, and the position of a gene on a chromosome is known as its locus. Okay, that's its physical location or its home. This individual, you notice, has 2 dominant alleles for the A gene. It's homozygous for the dominant allele. It's heterozygous for alleles at the B gene. It has a dominant and a recessive allele. And it has 2 recessive alleles for the C gene, and so it's homozygous for the C gene as well. Let's pay attention to some of these genes during the cell cycle. Here's an individual with 4 chromosomes. The A gene is on one chromosome, and both the B and the C gene are on the other chromosome. The genotype of this individual is heterozygous at all 3 genetic loci. It has 1 dominant and 1 recessive allele at each locus. After S phase, we completely duplicate the genetic information on one of the sister chromatids to the other sister chromatid. So we have a chromosome with 2 dominant alleles, another chromosome with the recessive allele, and this chromosome has the 2 dominant alleles, and this one has the 2 recessive alleles. The same exists at the end of G2, and after mitosis, we end up with 2 cells that have exactly the same genetic complement as the initial cell. Cytokinesis is the act of dividing the cytoplasm into 2 separate units equally. This is a frog egg, and you can see quite clearly the puckering and the folds where cytokinesis is initiated here as the cytoplasm is squeezed into 2 new daughter cells. This is a very short video showing cytokinesis in sea urchin. That's it. Let's look at it again. Nice and neat. Okay. Now here's another video showing cytokinesis in Chinese hamster ovary cells. Let's see, this takes a little bit longer here. Here's the condensed chromosomes. They're lining up. They're being captured by microtubules from the 2 centrosomes at the poles of the cells. Okay, now the cell's divided into 2, and you can see the chromatin is decondensing and a nuclear envelope is forming around it. Let's watch it one more time. Okay. There it goes. Mitosis and cytokinesis occur at the same time. Asexual reproduction is the process of making gametes. Parents make gametes with half the amount of the genetic material per nucleus. The gametes then fuse with each other to recreate the complement of DNA and chromosomes that are found in all the cells of the adult. Gametes are haploid, they're 1N, and somatic cells are diploid. They are 2 copies of each set of chromosomes. A diploid human cell contains 46 chromosomes. A sperm or egg contains only 23. During meiosis, you need to go from a 2N somatic cell to a haploid gamete. There are 2 successive divisions, Meiosis I and Meiosis II, but only 1 round of DNA replication. Now, most of the interesting events occur in Meiosis I. So at the beginning of Meiosis I, the cell looks very much like a regular dividing cell. It has 2 copies of each chromosome, and each chromosome has 2 sister chromatids. Okay, but the next thing that happens is not so the same as mitosis. It's quite different. What happens is that the sister chromatids line up and you exchange some genetic material. Then the nuclear envelope starts breaking down, and the bivalents, or the homologous pairs of chromosome, line up opposite each other on the metaphase plate. And microtubules attach to the chromosomes that consist of 2 sister chromatids together. And the chromosomes go to opposite poles of the cell. And then a cleavage furrow breaks the cell into 2, and now you have 2 haploid cells, 2 cells with only 1 copy of each chromosome. Notice that the centromeres do not separate, the sister chromatids do not separate from each other at meiosis I. That's one of the key features of meiosis I, is that you do not separate the 2 sister chromatids from each other. Instead, 1 whole chromosome of each pair goes to each pole. Okay, so the genetic exchange of material is a very important part of meiosis I. There's an actual physical exchange of chromosome pieces. The first thing that happens is that a synaptonemal complex forms with many very special proteins that line the 2 sequences of DNA up from sister chromatids of homologous chromosomes together. This is a very precise aligning of the DNA from the 2 chromatids. Then there are chiasmata that form, and an actual physical breakage and exchange. The chromosomes, when there are 4 sister chromatids for each of the 2 homologous chromosomes, it's called a tetrad. After the exchange occurs, the synaptonemal complex dissociates, and so the 2 homologous chromosomes can then go to opposite poles. So, metaphase of meiosis I, after the sister chromatid exchange, after genetic recombination and crossing over has already occurred, you need to separate the 2 chromosomes to make the cell a haploid instead of diploid. The bivalents are organized along the metaphase plate. Here they are, okay, opposite each other. The pairs of sister chromatids are aligned in a double row rather than a single row, as in mitosis. Okay. The arrangement is random with regard to the homologue. So this cell is showing that if all of these pink chromosomes came from mom, the assortment of that is going to be random. Some of the times you're going to get a mom chromosome, and some of the times you're going to get a dad chromosome, and that's completely random. One of the homologs is going to be captured by microtubules from one of the poles, and the other homologue is captured by microtubules from the other pole. And then the whole chromosome goes to one pole, and this chromosome will go to the opposite pole. The chromatids do not separate in meiosis I. They stay together. So meiosis II looks very much like mitosis. regular mitosis, where all the chromosomes line up with the centromeres and kinetochores right along the metaphase plate. At this point, the sister chromatids do come apart and they go to opposite poles, and now you have 4 haploid daughter cells with the chromosomes consisting of only 1 chromatid. So if you follow some genes along the process of mitosis and meiosis, you can see the genetics behind mitosis and meiosis. So this is very similar to what we already looked at for mitosis. We started out with an individual that was heterozygous at all 3 of these genetic loci, and we end up with 2 daughter cells that are also heterozygous at all 3 genetic loci. In meiosis, however, we have the same complement of genetic material at the beginning. This cell is also heterozygous at all 3 loci, big A, little a, big B, big C, little c. And it goes through the SG2 phase and duplicates the chromosomes so that each chromosome now has 2 chromatids. And then recombination occurs, and you cross over exchange some parts to it, so that now you have a chromosome with 2 chromatids, but this individual has a dominant allele for A and a recessive allele for A, whereas this majority of this chromosome up here had the 2 dominant alleles. So you've exchanged a piece of this chromosome. Okay? And for the B chromosome, B and C gene containing chromosome, you've also exchanged a little bit of DNA. Okay? So that this chromosome now has a big B allele and a little B allele, but it still has the 2 dominant alleles for the C locus. And when these 2 cells then divide and the chromatids separate from each other, you can end up with 4 different genetic compositions of each of these 4 cells after meiosis II. You'll have a big A, a little b, and a big C, etc., etc., etc. They're all different. In spermatogenesis, we can follow the progression of cells from a primary oocyte to the formation of mature sperm cells. And this closely mimics what happens in meiosis I and meiosis II. We start out with a diploid primary spermatocyte, and then we end up with, after meiosis II, 4 different cells that are now haploid, and then cellular differentiation occurs, and you end up with a mature sperm with a compact head and a long motile tail. In meiosis I, you'll end up with 2 haploid secondary spermatocytes. After meiosis II, you end up with 4 haploid spermatids. Okay? And then differentiation produces the mature sperm. Oogenesis is somewhat different. Okay? Unlike spermatogenesis, the divisions in oogenesis are asymmetric. In other words, when you have the nuclear division, one of the nuclei ends up with just a tiny little bit, and sometimes it's barely detectable, amount of cytoplasm around it. This is called the first polar body. And at the end of meiosis II, you end up with the second polar body. So oogenesis produces only 1 egg per primary oocyte, and that egg is haploid. You start out with a primary oocyte that's diploid, and you go through meiosis I and meiosis II to end up with a haploid egg, but only 1. That's because the egg needs to store a lot of material to sustain embryonic development through several divisions, cellular divisions, before the embryo can begin to extract nutrients from, in the case of placental animals, from the placenta of the mother. So in summary, diploid organisms have two sets of homologous chromosomes, one from the female parent and one from the male parent. Humans have 23 pair of homologous chromosomes, including the sex chromosomes. Genes are located at the same position or loci on homologous chromosomes. Chromosomes can consist of either one chromatid or two chromatids. The centromeres are comprised of DNA, and at the centromere can be found the kinetochore, a large complex of microtubules and motor proteins that separate the sister chromatids to opposite poles of the cell during mitosis. The cell cycle is divided into four main phases: G1, S, G2, and M. Cells in G0 are not dividing. During the S phase, new DNA is synthesized and chromosomes go from having one chromatid to having two sister chromatids. During mitosis, the chromosomes line up on the metaphase plate with the centromeres in the middle. Then the centromeres separate and the sister chromatids go to opposite poles of the cell. CYTOKINESIS is the process of dividing the cytoplasm into two new cells, each with its own nucleus and set of chromosomes. Meiosis is the genetic process of going from a diploid somatic cell to a haploid gamete. Meiosis has two rounds of cell division, but only one round of DNA replication. In meiosis I, the cell goes through G1S and G2 phases to produce bivalent chromosomes, each with two sister chromatids. Homologous chromosomes pair and crossing over occurs. At the cell division of meiosis I, the centromeres and sister chromatids do not separate. The sister chromatids remain attached. Each two-chromatid chromosome of a homologous pair goes to the opposite poles and into a new cell. In meiosis II, no new DNA synthesis occurs. The chromosomes line up at the metaphase plate, and then the centromeres separate. Each cell ends up with a single copy of each chromosome, which consists of one chromatid. Genetic recombination occurs at meiosis by two mechanisms. First, the chromosomes can randomly assort at the cell divisions, and second, crossing over or sister chromatid exchange can occur. Spermatogenesis yields 4 haploid sperm for every diploid primary spermatocyte, but oogenesis yields only one large egg cell per diploid primary oocyte. The rest of the genetic material is partitioned into polar bodies.